So we've talked about in the last video how Hardy-Weinberg works mathematically, but let's look at an example of how it can work in the real world. So there's a condition called PKU that is caused by a single gene locus, and it seems to meet all 5 of the Hardy-Weinberg assumptions. So first, it has a very, very low mutation rate, and so we can look at that as essentially 0. Mate selection is random, so we have random mating, essentially because you don't really know if somebody else has PKU. It's very treatable, and so if someone gets it, it tends not to be noticeable or cause any big impact. Because it's treatable with some simple dietary restrictions, natural selection doesn't have a big impact on those homozygous individuals who have that recessive condition. The population is large. Right, it's spread randomly around the global population, and there are a lot of people on the globe. And migration has no effect because it's not more common, for instance, in the US than it is in Asia or in Europe or in Africa. So let's see if we can do some math and figure out how many people are carrying this thing. So we know that about 1 out of every 10,000 people have PKU, right? And that is a phenotype, right? So they have the phenotype of PKU, and that said, because it's a recessive disorder, we know that if they have that phenotype, what their genotype will be, right? It will be homozygous recessive. And the frequency of homozygous recessive individuals is Q squared, so we know 1 out of 10,000, or 0.0001, is Q squared. Well, mathematically, where do we go from there? Well, we can get Q. Right? If we know Q squared is 0.0001, then Q is the square root of that, or 0.01. Well, then it's pretty easy. If we know Q, we can figure out P because P plus Q equals 1. So if Q equals 0.01, then P must equal 0.99, right? You just take 1 minus 0.01, you get P equals 0.99. And from there, it's pretty easy to get the genotype frequencies that we're missing. So 2PQ is 2 times 0.99 times 0.01, or 0.0198. And you can also get P squared if you want, right? Do 0.99 times 0.99. That would give you the frequency of homozygous dominant individuals that are not carriers. But 0.0198, these are heterozygotes. This tells you the number of people who are carriers in the U.S. population, about 2%. And it turns out that in genetic testing, that is roughly the case. So I mentioned that the 5 assumptions that you have to meet to be at Hardy-Weinberg equilibrium have to do with the 5 things that can cause you to evolve as a population. Now we're going to narrow that down to really 3 things, because one of those 5 assumptions was no mutation. But mutation is inherently a pretty random thing, and so there's generally no net mutation. In terms of random mating, one of those other assumptions, that has to do with sexual selection, which ties into natural selection. It's a type of natural selection. So we'll talk about that within that context. And in terms of large population size, right, we're going to talk about genetic drift, because genetic drift is actually what causes that evolution. Genetic drift, or the effect of randomness on allele frequencies, is just more pronounced in smaller populations. Natural selection is the most obvious one of these to see in the wild. It's also the only one that can cause adaptive evolution, right? For instance, genetic drift can cause change in allele frequencies, but it doesn't necessarily make a population more fit, more adapted to their environment, whereas natural selection does. One example of this is DDT. So DDT is a pesticide that was used frequently in the first half of the 20th century, first 2/3 really. And when exposed to DDC, there was a big selection pressure on fruit flies because it killed fruit flies. And fruit flies who were resistant in DDT did better. So there's a big selection pressure on fruit flies to be resistant to DDT. And over multiple generations, you found fruit flies that were significantly resistant to DDT when it was used in agriculture. Genetic drift, as I mentioned, has to do with just random fluctuations in gene frequency, allele frequency, from one generation to the next, right? And these random chances have a much greater impact on a small sample size. So in the last video, I used the example of a coin flip, right? You have 50% chance of getting heads, 50% chance of getting tails. And so if you flip it 10 times, you would expect 5 of each, but if you got 7 tails and 3 heads, that wouldn't be crazy, right? But if you flipped it 1,000 times and got the same frequency, 700 tails and 300 heads, then you would assume there's something wrong with your coin. So this is how that works. Here we have a small population of flowers, and remember we have the two same alleles we were talking about in the last video, right? CR for red, CW for white, color alleles, and if you're heterozygous, you are pink. And our P is 0.7, frequency of red alleles. Our Q, or frequency of white alleles, is 0.3. Now, if we let them reproduce to the next generation, it's very possible in this small population that fewer red flowers happen to leave offspring, right? Or the heterozygous flowers happen to leave more white offspring than red offspring or pink offspring. And anyway, our allele frequency changes to 0.5 and 0.5, entirely due to random chance. Now it can flip back in the next generation. It's hard to tell, but every now and then you might get something like this, where from 0.7, 0.3 to 0.5, 0.5, just by random chance in this small population, really only 2 plants leave offspring, and it happens to be the red ones. And the next generation only has the red allele. And from here, you can't go back, right? Short of gene flow, right, getting a white allele from another population, from here on until the end of this population, or maybe mutation, right, we are stuck with only red flowers. The red allele is what we call fixed in the population. And all of this is just due to random chance, to genetic drift. I recommend checking out this video here, Mechanisms of Evolution. You can find it in the A&P Bioflix. I believe it's also in one of the questions that I've assigned you on Mastering Biology. Now, there are a couple of situations where randomness and genetic drift can have a pronounced effect on natural populations, and one is the founder effect. So this occurs when you have a big population, a main population that's large, and a few individuals wind up colonizing a new habitat, and those new individuals may have a similar allele frequency to the original population, right? But they may not, right? It may just happen that you have a weird different allele frequency that lands on that new island, right? When those birds have been blown off course or whatever. And that new island population starts off with a completely different allele frequency than the original large population. This is called the founder effect. And that founding population is usually pretty small and so is pretty susceptible to genetic drift. And this is one way that when you get new small founded populations coming from a larger population, you can very quickly get some differences that begin to evolve in that population. A similar scenario comes with the bottleneck effect. So here, instead of starting with a large population and having a few that fly away and colonize a new island or a geographically isolated area and start their own population, as we get with the founder effect, we have a large population that, for one reason or another, has a sudden large population reduction, and you only have a few individuals left. So let's take a look at our gene pool in this situation, right? So here's our original population. Now, when you randomly figure out which individuals survive that large event, they may or may not represent what allele frequencies you saw in the original population, right? In this case, our surviving population looks significantly different. There are more blue individuals here than there were in the original population. There are no yellow individuals left in the surviving population. And this surviving population is small and so more susceptible to genetic drift. Now, the surviving population may grow and get larger and become as big as the original population was, but its allele frequency is forever changed. And this is important because it can help us understand how our activities, which can sometimes cause these large population drops, can affect other species. One example of this is the cheetah. Cheetahs have a pretty healthy population now, but they had a relatively large die-off as a result of human activity in the past century. Before conservation measures were taken, and then they increased in population size. But they are almost clones of one another. There is very little genetic diversity amongst cheetahs, right? And that's because of that big bottleneck. And so, again, understanding the effects of this lack of genetic diversity and where it comes from with these bottlenecks can help us manage these populations when we talk about conservation ecology, which we'll get to at the end of the semester. Another example of this is the greater prairie chicken. And the greater prairie chicken used to live in all different kinds of places in the Midwest, but there's been a severe loss of prairie over the last many, many decades. And so that loss of habitat has led to a loss of birds, and that bottleneck led to a lack of genetic variation. And that lack of genetic variation meant that they suffered some deleterious effects. They lost some good alleles, and their egg hatch rate went down to 50%. Now, this was a big problem for this Illinois population of the greater prairie chicken, which again had lost significant amounts of their range and suffered a bottleneck in population. But in other populations, Kansas and Nebraska, that had no bottleneck, they didn't have the same lack of genetic diversity, and they didn't have the same issue with percentage of eggs hatched. And so conservation biologists took some individuals from Kansas and Nebraska, allowed them to breed with the Illinois population, and get a little bit more genetic diversity in there. And once those different chickens added genetic diversity to the Illinois population, the egg hatch rate went up from 50% to 90%. And this was sort of a big example of a success in conservation biology using our knowledge of bottlenecks and genetic diversity. So as a quick summary, genetic drift is significant primarily in small populations. It can cause allele frequencies to change at random. That can lead to a loss of genetic diversity, and sometimes harmful alleles can become fixed in the population because you've lost all other possible alleles. Our next driver of evolution in populations is gene flow, or the movement of alleles between populations. And this can happen when fertile individuals end up migrating out of one habitat into another, or between one population and another. And this can affect the fitness of a population either positively or negatively. So in this case, it's going to impact the population negatively. So there are genes or alleles that have become common in island populations of this particular bird, the gray tit, and they make the population more fit on the island, but less fit on the mainland. And so when birds come from the mainland to the island and bring those alleles with them, the island individuals, especially in the place, in the central population, where you get the most migration from the mainland, you get a drop in fitness for those populations. That said, that gene flow can also bring genes that are beneficial, right? So in this case, the spread of alleles for resistance to insecticides. So we've been using insecticides to target mosquitoes that carry the West Nile virus and malaria. And in some populations, those mosquitoes have evolved resistance. And when those individuals move into other populations, they bring those resistant alleles with them. And while that may not be positive for us at all, it is positive in terms of the fitness of those populations of mosquitoes. So, genetic drift has a random impact on a population. It could be positive, negative, or neutral. Gene flow could have a positive or a negative impact on a population. That means that the only driver of evolution that consistently brings adaptive evolution is natural selection. In the next video, we're going to talk about a few ways in which natural selection can drive allele frequencies in a population.